Okay. See, nucleic acid blotting techniques. Blotting techniques are very widely used analytical tools for specific identification of desired DNA or RNA fragments. That means within the nucleus, we have so much of DNA, RNA, no? So if we want to just identify and study a particular part of DNA fragment and RNA fragment, we generally use this blotting techniques. Okay. So
Yes, ma'am.
blotting means like process of immobilization. Just a minute, beta. So process of immobilization of sample nucleic acid on solid support. That means when you take a solid support and try to just get nucleic acid piece to be deposited on the solid support, we will be able to study that particular part of DNA or RNA. That means sequence of these genetic code, like whether it is ATG, CTG, what is there exactly in that particular fragment of DNA. We are just cutting and we are trying to just get it fixed on a solid support. For that, we generally use like nitrocellulose or nylon membranes. The blotted nucleic acid, that means solid support is nitrocellulose beta or nylon membranes. The blotted nucleic acids are then used as targets.
Yes, ma'am.
Hybridization experiments for their specific detection. So the outline of nucleic acid blotting technique is depicted in this 27.6 picture beta. Let us see this one. Immobilization. We are just trying to first stop the mobility of either the fragment of DNA or RNA. So in order to stop the mobilization of DNA, we use southern blot, RNA, northern blot. Both DNA or RNA, we can use dot blot. Okay. So then pre-hybridization is done. That means what? We have just cut a piece of DNA that is labeled either DNA or RNA. You just name it like aDNA, bDNA, aRNA, rRNA. You call it as labeling. So labeled DNA or RNA probes. Then before hybridization, you call it as pre-hybridization followed by hybridization. Then it is followed by series of washes, which is called as stringency washes, and then we will be detecting with special devices that particular fragment of DNA. Okay, so that is actually a diagrammatic representation of typical blotting apparatus. It looks like this. Okay, you just try to learn as many pictures as possible, beta. Like there is a support block, there is a gel, filter paper membrane, paper tissue, glass plate, weight. All these we use generally to just fix the fragment of DNA RNA hybridization. Now in this southern blotting technique, we generally hybridize what? DNA, no? So suppose this is nucleus. So in the nucleus, we have so much of genetic material DNA. We are only taking a fragment of this genomic DNA. We are like, first of all, the DNA is cut into fragments, that is pieces. Then it is allowed to undergo gel electrophoresis. Electrophoresis means you are passing the electricity in a solution. Okay. So when you are allowing it to get fixed. Nitrocellulose paper, just now you have studied, isn't it? Solid paper should be. So you are allowing these DNA fragments to move, mobilizing it, and we are fixing it on a paper so that it is immobilized forever. Okay, so you are finding at one end long DNA fragments are fixed and at the other end short DNA fragments are fixed. Okay, we are making use of agar rose gel. Okay, agar is a seaweed. Then denaturation by mild alkali. Okay, which is actually called as Biot transfer. So initially electrophoresis is done on agar rose gel beta, followed by you're transferring these fragments of DNA into nitrocellulose so that you will be able to fix in it and study about that fragment. Okay. So now we are making use of DNA probes. Probes will study the genetic information. What is the sequence of nucleic acid which is present in the fragment of DNA which you have cut and separated. Okay, so these are hybridized bands Bands are nothing but the DNAs only. When probe is allowed to just study this, you will see they will be shining like this, and we will be able to just read the sequence of the genetic information with the help of a machine called as autoradiograph. So this technique is southern blotting technique. Okay. So this southern blotting technique starts here, beta. Just the help of that flow chart, you will be able to learn very easily. Okay.
Yes, ma'am.
So southern blotting technique is named after the scientist Southern 1975 who developed it. Other name is northern and western blotting, which are now accepted. Western blotting involves the transfer of protein blots and their identification by using specific antibodies. So we have used this flow chart already. Southern blotting also, did we study this? 27.6 we did. 27.7. Yeah, this one. So but just look at this flow chart, beta, and just read it once, you will understand. Okay.
Yes, ma'am.
Why do we use the southern blotting technique? So they are very much sensitive, although it is a simple technique. So let us read, like, why do we use? It is an invaluable method in gene analysis. So suppose we don't know if the baby is really belonging to a particular father, then we can take a particular fragment of DNA from the father and also from the baby, and we will try to match it, no? So that is invaluable method in gene analysis for confirmation of DNA cloning results. So like forensically, we usually apply more to detect minute quantities of DNA, whether it is in the hair, nail, okay, whatever thing we find at the crime site, we just collect it and we will just check with the help of this southern blot and northern blot techniques to check if the sequence of that genetic information what we got from the crime scene, is it similar to the one what the suspect has. Okay, so that is to identify parenthood, thieves, rapists, etc. Okay. So highly useful for the determination of restriction fragment length polymerism. This is what you have written once. No, but RFLP. You have asked this doubt also. For the
Yes, ma'am.
determination of restriction fragment length polymerism. We'll see this also in detail, associated with pathological conditions. That means to determine if the person is suffering with any disease, that is RFLP. Okay. Then we have another technique which is called as northern blotting method. Why do we check this? For the identification of which molecule? RNA.
RNA.
Yeah, RNA. So this is RNA extract. Okay. Same like how we have extracted DNA. Now we are extracting RNA. Now again we are allowing it to just fix it in agar-rose gel. There also we have used. And we are undertaking electrophoresis. You get RNA bands and now you are hybridizing. What are we using? Nitrocellulose, isn't it? You're just fixing it there. With the help of DNA probe, we are able to study the sequence of nucleic acid present in the RNA. So this is an outline of northern blotting also. Okay, beta?
Yes, ma'am.
Then southern dot blotting is modification of southern and northern blotting techniques. So both DNA or RNA directly spotted in filters. That means if we want to study both, we can go with this technique. Okay, under which we have western blotting. So we are able to identify any proteins. Okay, whether it be DNA, RNA, immunoglobulins, anything can be studied with the help of western blotting. Okay, all proteins. Useful to understand the nucleic acid functions, particularly during the course of gene manipulations. Same thing, but a technique of western blotting is involving transfer of electrophoresed protein, in the sense agar-rose gel, we are undertaking electrophoresis. It can be DNA or it can be RNA or any other protein. These protein bands from here, they are shifted to like polyacrylamide gel or nylon or nitrocellulose membrane, isn't it? So these proteins can be detected by specific protein ligand interactions. So we can study antibodies, lectins, okay? So autoradiography, I said it's a machine which is actually in the first picture, when we have studied Western blotting, here only we got this term autoradiograph, which is a machine which is actually helping us to detect the sequence of nucleic acids with the help of probes. That is autoradiography. Okay.
Yes,
Localization
ma'am.
and recording of radio label within a solid specimen. What is a solid specimen here? Nitrocellulose, no better. Polyacrylamide
Yes, ma'am.
or nitrocellulose is the
Nitrogen.
membrane, isn't it? Over which what is there? Either DNA fragment or RNA fragment or a protein. With the help of probe, we are reading the sequence of nucleic acids or amino acids. Okay, that's how we are able to read the sequence. Are you understanding better?
Yes, ma'am.
Okay, then applications, that whatever we have studied under southern blotting, but the same thing is application for everything. Northern blotting, western blotting, everything. Okay.
Yes, ma'am.
उसके बाद northern blotting, western southern, RFLP, PCR. See, probes are these like nothing but it's see, when you just go to western blotting, so here what is the word which is used after nitrocellulose, you are finding the bands of DNA, right? So the
Yes,
DNA
ma'am.
bands are actually studied with the help of probe. Probe in the sense like probes hybridize with complementary DNA molecules on the paper. You remember tRNA has like anticodon site.
Yes, ma'am.
So suppose AUG is there, correspondingly for AT will be there. U
C will be there.
C, isn't it like that?
C,
That means
ma'am.
we have complementary base pairing. Similarly, probe
Yes, ma'am.
also will have something like complementary base pairing type of information, with the help of which we will be able to study the sequence of nucleic acid in the fragments of DNA, RNA, or proteins. Okay, better. So those are
Yes, ma'am.
probes. They hybridize with complementary DNA molecules on the paper. Okay, that is the meaning of probe. Same like anticodon area site, like that it has function. Recombinant vaccines. Now we have to check out. This is important button. What is the importance of rDNA technology? What is the disease and oh, not this one, not this. This is general thing. Anyhow, you study in second year microbiology. RFLP, it has come in detail, we will study. DNA fingerprinting is the same thing, madam. Beta, meaning what do you understand is same like the fragments of DNA are selected and we are reading the sequence of it. We are trying to match with the culprit.
Yes.
Is it the same? Suppose
Yes,
you
ma'am.
get a hair or you get the dried semen spots over there or something like that, like a crime scene or blood spots or whatever. You are collecting the same thing from the culprit, whom you are suspecting, and just trying to match whether it is the same DNA or not. That is what is
Or
called
not.
DNA fingerprinting. Okay.
Yes, ma'am.
Now let us see this like restriction fragment length polymorphism, RFLPs. They represent stretch of DNA that serves as marker for mapping a specified gene. They are located randomly throughout person's chromosome and have no apparent function. So a DNA molecule can be cut into different pieces by a group of enzymes called restriction endonucleases. So these fragments are called polymorphism, literally means many forms. So outline, let us see. First see, we will go with the previous picture. Then we'll come to this. .22. First picture, what we are asked here, 27.1. Oh, this is stable. Let's this we know, no, beta. We just cut a fragment of DNA. You said you know that flow chart at the beginning of the
Yes,
chapter.
ma'am.
Restriction.
Recombinant technology. Yes, ma'am.
Using, and we are cutting a fragment of DNA. Okay. Now after that,
Yes, ma'am.
what are we doing? So we are trying to mark a specified gene. So let us see how do we do that. See here, an outline of restriction fragment length polymorphism. You have R1, R2, R3. This is one DNA. You are making use of restriction endonuclease, and this DNA one is now cut into 4 fragments. Okay. So R1 and R3. Now R2 you have selected and it is being removed. And the remaining are like R1 and R3, they are just pasted back. Okay. So now this is, initially it is called as DNA 1. Now it is DNA 2. Okay, again you are making use of restriction nucleus, and again you are cutting into fragments and you are choosing whatever part of DNA you are trying to study. Okay, so that is restriction fragment length. So lengthwise you are keeping on cutting, but restriction fragment length polymorphism. Okay.
Yes, ma'am.
So you, with the help of this diagram, and you just read these points, you will definitely understand. Okay. So why do we use this? We will be able to diagnose certain diseases. Okay, where if there is any defective gene is there, we are trying to trace with the help of this technique, RFLP. Okay. We have many genetic disorders, no? Like Down syndrome is there, Klinefelter syndrome is there, sickle cell anemia. All these are genetic disorders which can be like studied with the help of this technique. Okay, beta?
Yes, ma'am.
We have to go with PCR, isn't it?
PCR, ma'am. Yeah.
Here we are. See, polymerase chain reaction. It like polymerase chain reaction is nothing but synthesis of protein in vitro conditions. But in the lab, no, we are providing all amino acids, required enzymes, a particular temperature, pH, and all. There will be an automatic machine which will be allowing the amino acids in between which peptide bonds will be formed and polypeptide chain will be formed, and it will be like whatever desired protein is there, we are synthesizing in the laboratory conditions, that is in vitro. That is the meaning of polymerase chain reaction. Okay, now RFLPs can also be like scored by this technique for this purpose. But actually there is nice flow chart, but I remember in some other chapter, I guess, not this chapter at all, or let us see, because what is the last page given for PCR?
I'll check, ma'am.
From the index. Is it the same edition both of us are using?
Yes, ma'am.
Polymerase chain reaction is a laboratory, I said, in vitro technique for generating large quantities of a specified DNA. You are synthesizing now in the lab better. Okay, obviously PCR is cell-free. It is not inside any living cell. It is in the laboratory. And it is multiplication technique. That's what is called as amplification technique for the synthesis of multiple identical copies of any DNA of interest. Okay, that is the meaning of PCR. Then, so principles, what is like a double-stranded DNA of interest, like you make use of restriction endonuclease to cut a fragment of DNA, right? Here also we are doing the same thing. Double-stranded DNA of interest is denatured to separate. Denatured in the sense the bonds are broken and the strands are separated. Understood better?
Yes, ma'am.
Two individual strands. Each strand is then allowed to hybridize with a primer. Okay, that I'll show you. Okay, the primer template duplex is used for DNA synthesis. These 3 steps, like we have denaturation, renaturation, and synthesis. They are repeated again and again, and you can actually generate as many DNA copies as you want. So from here till here you keep. Technique we will study with the help of flowchart, and just give one reading. You will be able to write in your own words. Let us see the technique now. See, this is the technique what we are using. This one. See, the polymerase chain reaction representing initial 3 cycles. First thing is denaturation. This is double-stranded DNA, Shubhangi, 2 black lines. Okay,
Yes, ma'am.
you are just exposed to some chemical agent, or you have heated it, or doing some thing which is now leading to separation of 2 strands. Can you see both are separate now? This
Both are separate.
is primer. You remember primer also has come when we were doing replication.
Replication, yes, ma'am.
इधर से दो तरफ से बाइंडिंग होता और डॉटर स्ट्रैंड बन जाता ना. Similarly.
Yes, ma'am.
So now see DNA synthesis. See, this is the original strand. This is also original strand. ये वाले दो primers हैं ना. Primers same वो जो natural technique है वही आप artificially you are using it to synthesize the daughter strands. Okay. So this is long template. This is also long template, which is formed complementary to the information which is present in the parent strand. Okay, beta. Then
Yes,
denaturation, annealing, synthesis. So it goes stepwise. यहाँ पे देखो दो डॉटर डीएनए फॉर्म हुए, फिर आप क्या करें, फिर उसको अगेन यू हैव denatured, अगेन यू हैव गिवन more primers, that is annealing, again you have seen more daughter strands are formed. That means it's going on multiplying and multiplying. अब यहीं पे अगर अपन देखें तो 1 2 3 4 copies of DNA have formed here, isn't it?
Yes, ma'am.
So along with original strand, you are finding 2 long templates were there. Now these 2 long templates will act as the original strand. Then again primer will come and the base pairing starts. Again you go with denaturation, annealing, synthesis. See how many DNA have formed copies 1, 2, 3, 4, 5, 6, 7, 8. As many as you want. That machine will help you to synthesize the copies of DNA. That's what is polymerase chain reaction. Is it clear, beta?
Yes, ma'am.
So you just read the technique with the help of the picture what is given here, what I have shown you just now. Okay, 27.16. Then you will be able to write it very easily. Then sources. That's it, beta. इतना ही है और कुछ नहीं है इसमें.
हाँ, ma'am.
So